The S phase of the cell cycle is so named because it represents the time in the cell's life when DNA replication takes place. The synthesis phase is carefully placed between two gap phases or growth phases, allowing the cell to complete enough growth to do DNA replication, and then allowing the cell to complete enough growth to complete mitosis or meiosis, depending on what type of cell it is. In the S phase, what happens is that each chromosome or each piece of DNA, and that's what we have here, this is a chromosome, each piece of DNA becomes unwound and a copy gets made, an identical copy. Strangely enough, though, the identical copy stays attached to the original copy at the centromere. The centromere is just an attachment point. You can kind of think of it like the waist. And when I mean waist, I mean like W-A-I-S-T, like I wear a belt around my waist. Also recall that chromosomes could possibly be the same length, but they had have their centromeres placed at a different area. So that would make them different chromosomes. Take a look at these two pictures here in the gap 1 phase and in the gap 2 phase. It's very subtle, but you can see that all of the chromosomes have been duplicated and the identical copy is attached to the original. And I want to impart something that is very important to you, that if you ever want to count chromosomes in a cell, and this is important, if you ever want to count the number of chromosomes in a cell, the only thing that you can do reliably is count the number of centromeres that you see, because this over here is one chromosome. This over here, this is also one chromosome. It just has two copies of it. As with many processes in the body, as with all of them, DNA replication is done by an enzyme, and this enzyme is called DNA polymerase. The ending ASE indicates to us that this is an enzyme. That's a common ending for enzymes. And this is the enzyme that makes the DNA polymer. And so it knows the base pairing rules that cytosine adds or matches with guanine and adenine matches with thymine. So when it encounters a guanine, it will look for a matching cytosine. When it encounters an adenine, it will look for a matching thymine and opposite. I can never find an appropriate picture of DNA replication that suits my preferences, so I made this awkward picture here. That's me. I made that awkward picture here. What you can see is that I am showing you one length of DNA, and it has a centromere. And what I'm showing you is that the DNA is in those two strands, right, making that double helix, and that each one of those strands becomes replicated by DNA polymerase. So what ends up getting produced is this very large structure on the right, which again, this is one chromosome. It is just in its duplicated state. I know that you might be arguing with me and saying, no, Amy, that's two chromosomes, that is not one, and it's duplicated. I know, and this is what becomes very tricky for students, because the previous slide in this mini lecture said that if you want to know how many chromosomes, I would count the centromeres. So again, there's just one centromere here, and therefore that is one chromosome. DNA replication is known as semi-conservative. I want you to compare the picture number 1 on the top left to picture number 6 on the bottom right of this slide. Notice how on the bottom right we have 2 new strands of DNA, right? And these 2 new strands will get folded up into their double helix state. But we have 2 new strands of DNA, which are orange, and then we have 2 old strands of DNA, which are the blue. So each one of these new DNA molecules contains one old or original DNA strand and one completely new strand of DNA. And this is what semiconservative means. It means that we are conserving 50% of the original DNA into each newly synthesized DNA molecule. This is different than non-conservative, in which we would make 2 completely brand new molecules and throw away the original. This is also different than completely conservative, where we would maintain our old copy of DNA intact and make one completely new copy of DNA. Semi-conservative is like the best of both worlds, that you get to retain some, but you also get to make some new DNA. As you saw from the previous slide, all of the steps between pictures 1 and 6 are pretty intense. And this is true, but that level of detail is also more than what we need for this class. In another class that I teach, we take a look at a diagram such as this, and this diagram shows you that it's not just DNA polymerase that acts in DNA replication. There are enzymes like DNA helicase, which breaks the hydrogen bonds between the DNA strands. There are molecules or enzymes called topoisomerase, which stabilizes the unwinding DNA. And then there are various versions of DNA polymerase, and you can see pol 1 and pol 3 here. Also note that DNA replication on the top strand is occurring in an opposite direction than it is on the bottom strand there. And this is true, that DNA replication actually involves 2 very different ways to synthesize new DNA. Just a reminder that when you make DNA, you can't just let the double helix hang out in the nucleus. You have to condense the DNA back into a nicely packaged chromosome. And to do that, you first need those histones around which we're going to wind the DNA. And then we're going to make nucleosomes, which further package the DNA. And then these nucleosomes are all going to be coiled up into that structure of DNA that we call the supercoil, which basically packages it into a chromosome. This type of packaging, this type with histones and nucleosomes, is only present in complex organisms called eukaryotic organisms. Bacteria that get all of the needed events of life done don't have this complicated packaging of their one chromosome. I just always want to take the opportunity to remind you that bacteria does with one chromosome what it takes us humans 46 chromosomes to get done. I mean, bacteria is so awesome. It's like, I don't even need to keep a copy of this one chromosome anywhere because I'm not going to ruin it. Whereas humans are like, any one of my chromosomes could get ruined any day now. So I just have this extra one just in case.